Okay, so welcome back to this next video in which we are discussing circadian rhythms. Okay, so we've discussed that a circadian rhythm is some process which repeats on a 24-hour cycle, basically. And we've discussed many physiological examples now of circadian rhythms. We've also seen the example of the mimosa plant, which has this circadian rhythm which involves moving its leaves up and down on a 24-hour basis. It moves them up during the daytime and then down at the night time. And everyone thought that this would be a response to the light level, i.e. that when the light was present, the plant would respond by moving its leaves upwards, and then when the light diminished, it would move them back down again. Okay, that would make sense. However, there's this nonsensical sensical answer, which is that basically, even if you take the mimosa plant and put it in a darkened room, it will continue to move its leaves up and down on a 24-hour cycle, even if you remove the light, basically. Okay, so the plant seems to have ways of telling the time that don't rely on the light. And this shocked people. The first person who discovered this thought that the plant must be sensing day and night through some other means, basically, potentially through the humidity of the air or something along those lines. He didn't believe that the plant could be telling the time. He didn't really see what the purpose of the plant being able to tell the time would be. Okay, why wouldn't it just move its leaves up in the response to light and then move them down in response to the dark? Okay, but that's the result. Now, we can do a similar thing in humans, basically. We've seen that there are all these physiological processes, such as us being awake or asleep, our core body temperature, our growth hormone, and our cortisol levels that are fluctuating on these 24-hour cycles. And you might think that they are all governed, surely, by the external light, by the circadian rhythm of the Earth spinning on its axis, and the experience of daytime and nighttime that therefore produces. Okay. However, what you can do is you can put a human in a cave, okay, where there are no ways that he can tell, or he or she, I think we have used men mainly, that they can tell whether it is nighttime or daytime, basically. Okay. So firstly, let's just discuss a little bit of terminology. Okay, so I want to just discuss the terminology of what is meant by a Zeitgeber. People often use this term, and what it means is literally, in German, it translates to time giver. Okay, and it refers to things which allow you to infer the time of day, basically. Okay, so these allow you to understand what time of day it is. So, for instance, the light-dark cycle is a Zeitgeber because it tells you what time of the day it is. Okay, so these are external stimuli which give you information about what time of day it is. Okay, so they're effectively external clocks rather than internal clocks. Okay, so what we're going to do is we're going to take a human, we're going to put them in a cave where there are no Zeitgebers. There's no way that they can tell what time of day it is because, frankly, nothing changes depending on what time of day it is. Their conditions remain exactly the same. And basically, you might expect that everything will mess up. However, things don't mess up as much as you'd think. Instead, their core body temperature and other physiological variables continue cycling on a 24-hour basis exactly the same, basically. Okay, so 24-hour cycling continues, basically, for body temperature. However, it's slightly different for variables such as sleep and wake. Okay, so the sleep-wake cycle, initially, if you put someone in a cave, they initially stay pretty close to a 24-hour cycle. However, if they stay there for weeks and weeks, their sleep-wake cycles become very, very odd. They become prolonged, basically. Okay, and this is what's known as free running. Okay, so we're going to make their environment devoid of Zeitgebers, and now their physiological systems will be free running, i.e. they have only got their internal clock to guide them on what time of day it is. And basically they tend to adopt sleep-wake cycles that repeat with a period of around 30 to 36 hours, which is quite surprising. Okay, so let me emphasize this by showing you what's happening here. Okay, so if we draw one of these graphs where we have time on the x-axis, and now we'll have to extend it, so we're going to need more than just 24 hours now. Okay, so it's 0, 6, 12, 18, 24, and this is real time. This is the time on the external world, basically. Okay, then we've got 30, and then if we go further, we'll have 36, then 42, and then finally all the way up to 48. So basically, what happens is that often they adopt cycles with regards to sleep and wake that have a period of around 32 hours. And generally what happens is they will stay awake for 20 hours on average, and then they'll sleep for 12 hours. So let me show this. Now, of course, this is going to become completely out of sync with real time, okay, because they won't stick to a period, and let me explain what I mean by that. So let's say initially they start off sleeping between 0 and 12 o'clock. Okay, then they go into the awake state, so this is the sleeping state. Okay, and this is the awake state here. And then they'll stay awake for 20 hours. So let's put that on here. So 20 plus 12 will take us to 32, which is somewhere over here. So they'll stay awake all that time, and then they'll go to sleep again. Okay, then they'll stay asleep for 12 hours, which will take us up to 42. Okay, so not 42, 44. Okay, and then they'll awaken again. So basically they're now completely out of sync, because initially they went to sleep at midnight. Okay, this was midnight real time. Now they're going to sleep at 44 hours, which is effectively 8 o'clock in the night for real time. And then, of course, the next time it will have gone out of sync again. So everything's going out of sync with the real world, basically. Okay, in addition, even though their sleep-wake cycle is turned to something like this, their body temperature continues to cycle on a 24-hour cycle. Now remember, body temperature and the sleep-wake cycle were perfectly sort of synchronized to the 24-hour clock when you had Zeitgebers, such as day and night, which were telling you when to go to sleep and when to stay awake. Okay, and that meant that you could have a good night's sleep, basically. That's very, very healthy to have your body temperature going down to 36 whilst you're asleep, and then going up to 37 whilst you're awake. These people in the caves aren't going to have that luxury. Okay, so if we now draw body temperature underneath, okay, and we'll do it over the 48-hour period as well. So 0, 6, 12 hours, 18 hours, 24 hours, 30 hours, 36 hours, 42 hours, and then 48 hours. Okay, and we'll have body temperature which fluctuates between 36 and 37 degrees Celsius. Then body temperature might still be fluctuating like so. Okay, so it's at 36 between 0 and 6 o'clock. Then it goes up to 37. Okay, and then it'll stay up there till you go, usually when you would go to sleep at around 10 o'clock, and then it'll be low all the way through to here. Then it'll go up again, okay, at 30 hours, and then it will go down again over here or somewhere like that. Okay, and you can see now that it's fluctuating out of synchrony with the sleep-wake cycle, and this will lead to a bad night's sleep, basically. So it's not healthy to do this. Often the people who do go down caves and do these isolation experiments end up going mad, basically. They end up, taking solace in the company of mice and things like that. Okay, so it's not a healthy experiment to do. Right, and it's believed that this lack of synchrony between things like the temperature cycle and the sleep-wake cycle is responsible for the side effects that doing an experiment like this has. Okay? In addition, what does this experiment actually show us? Well, it shows us that the regulation of the sleep-wake cycle is different from the regulation of body temperature and other physiological parameters which continue to cycle on 24 hours even though you no longer have the day-night cycle, and even though your sleep-wake cycle isn't actually fluctuating on 24 hours anymore. Okay, so what that shows us is that these variables such as body temperature, growth hormone level and cortisol level, they weren't actually being regulated by you going to sleep basically. They were on a circadian rhythm on themselves, and that's another quite surprising result basically. Okay, so what we now want to discuss then is the circadian rhythms of these physiological variables, okay, because those are reasonably well understood, whereas regulation of sleep and wake is actually really poorly understood. So we're going to concentrate on the regulation of the physiological variables and the neural correlate of an internal clock, okay. So the neural correlate of the internal clock, which controls the physiological variables but isn't so involved in controlling sleep and wake, because the one thing this experiment highlights is that there are two clocks basically, one that's regulating the sleep-wake cycle and one that's regulating the physiological variables. We're going to focus on the internal clock that modulates the physiological variables, because that one is well understood basically. And the physiological clock, which modulates these physiological variables, is something called the suprachiasmatic nucleus. Okay, and there are actually two suprachiasmatic nucleuses, so we should call them the suprachiasmatic nuclei. Okay, so I want to now tell you about these nuclei. So we're going to start off with their position basically. They are within the hypothalamus. So let's do some neuroanatomy then. So we'll draw the brainstem initially. So here is the spinal cord, here. Okay, then above the spinal cord you have the medulla, which is the first portion of the brainstem. So once the spinal cord has come through foramen magnum, the first portion of the brainstem is then called the medulla. Its long name is the medulla oblongata. Okay, so this represents foramen magnum, which is that large hole in the base of the skull. Okay, then you've got the spinal cord below that, and I think I'll colour this picture in to try and make it look simpler. Okay, right, so let's colour in the different portions. So we'll have the spinal cord in green here. Okay, so this is the spinal cord down here. Then, once you've gone through foramen magnum, which we'll colour in pink here, you then go into this portion of the brainstem known as the medulla. Okay, so I'll colour in the medulla in blue here. Okay, so this is the medulla. Full name medulla oblongata, but we'll just call it the medulla. Then above the medulla you have the next portion of the brainstem, which is the pons. Okay, so this is the pons. And then above that, you then have the midbrain. Okay, so above the pons, we then have the midbrain. So I've coloured in the pons in orange, and then here we have the midbrain. Now, the suprachiasmatic nuclei aren't going to be in the brainstem, they're going to be in the hypothalamus, but I'm going to show you where the hypothalamus is relative to the other neural structures, basically. So here is the midbrain in purple here. Okay, now, so this is going up towards the top of the brain. The cerebral hemispheres will be here, basically, sitting alongside. The cerebellum will be behind here. Okay, then what we have sitting on top of the midbrain is two thalami. Now, because we're looking from a side aspect, we're only going to see one of these thalami. We're going to see the left thalamus because we're looking from the left-hand side. Okay, so the thalamus is this sort of egg-shaped structure that sits on top of the midbrain. Okay, and you have one on either side, a left thalamus and a right thalamus. Okay, then in front of the thalamus here, what you have is a structure called the hypothalamus. Okay, and this sits here, and it's got a few landmarks, one of which is the mammillary bodies, and again there's a left mammillary body and a right mammillary body. We're only seeing one here. Okay, so this is the mammillary body, and they're extremely important in memory. Okay, so if you obliterate those, it produces diencephalic amnesia. Okay, so this is the mammillary body, and that's the left mammillary body again. Then, in the middle, we have the pituitary stalk coming down, which will then have the pituitary gland below here. Okay, so that's the pituitary stalk with the pituitary gland. So this is the pituitary gland. And then at the front, we have the optic chiasm underneath the hypothalamus here. So this is the optic chiasm. So this is the entire structure shown from the side. And what I now want to do is draw another picture where we're looking down from above. So instead of looking from the side now, let's look down from above and see what we'll see. So we'll now see the two thalami, basically. Okay, and I'll draw these at the back here, or up here. Okay, so we're looking at these two structures. Now this is the right thalamus, this is the left thalamus. So this is this here. So I'll highlight it in to show you what's analogous to what. So this portion here is this here. This is the left thalamus, and here is the right thalamus, which we couldn't previously see on this picture. Okay, and there's a space in between them, and I'll tell you what that space is called in a moment. The hypothalamus is then this sheet that also goes around like so. Okay, so we're seeing the left side of the hypothalamus here. Okay, but there is also a right side here. Okay, so this is the right side of the hypothalamus, this is the left side. So there's a sort of cavity in the middle between the left side of the hypothalamus and then the right side of the hypothalamus here. Okay, now this cavity that's between the two thalami and also between the two halves of the hypothalamus, okay, this is known as the third ventricle. Okay, and it's full of cerebrospinal fluid. So this is the third ventricle. And then it is continuous with the fourth ventricle below over here that's on the back of the pons, because going through the midbrain you have a little channel called the cerebral aqueduct, which then takes the cerebrospinal fluid into the fourth ventricle, which is sitting at the back of the pons here. And then the fourth ventricle is then continuous with the central canal, which goes all the way down the spinal cord, basically. Okay, so the third ventricle then is full of cerebrospinal fluid, so this contains CSF. Okay, so the suprachiasmatic nuclei are within the hypothalamus, so where are they positioned on this structure here? Well, again, you have a left suprachiasmatic nucleus and a right suprachiasmatic nucleus. And they're in this sort of position here when we look at a picture like so. Okay, so this would be the left suprachiasmatic nucleus on the left-hand side, and this would be the right suprachiasmatic nucleus on the right-hand side. And the entire thing here, I should have just labelled this up, is called the hypothalamus. Okay, right. Now, if we draw them on this picture, of course, we're only going to see the left suprachiasmatic nucleus if we draw it on this picture, because we're looking at the left-hand side, but it would be in this sort of a position here. Okay, so let me highlight that up. So in blue here, this is the left suprachiasmatic nucleus, and you can see now why it's called the suprachiasmatic nucleus, because it sits nicely above the optic chiasm, basically. So that's why it's called the suprachiasmatic nucleus. Okay, let's just draw one more picture to show the anatomy here. And let's this time look from the front, okay? And then we'll see the two suprachiasmatic nuclei together at the front. Okay, right, so I need another piece of paper. Or in fact, I could probably squeeze it in here. Okay, so let's say this is the hypothalamus here. Okay, this is the front of the hypothalamus that we would be seeing here from above. Okay, so effectively, this line here that I'm now colouring in pink is this line here, okay, that we were seeing from above. Okay, then what we've got is the optic chiasm at the bottom here. So this is the optic chiasm with the optic tracts going backwards like so, and then the optic nerves coming forward. Okay, like so. So these two nerves that are going backwards, these are the optic tracts. Okay, and they'll be carrying visual information back to the lateral geniculate body. Okay, and then we have the optic nerves here, which are coming from the eye. Okay, so this is coming anteriorly, basically. So this is an optic nerve. This specifically is the right optic nerve here, and this is the left optic nerve. And this structure here, where they're crossing over, is the optic chiasm. Okay, so down the middle of the hypothalamus that you can't see, you have the third ventricle. Okay, so I'll draw that in dotted lines. You obviously can't see that because there is this portion that sits in front of that. And then the suprachiasmatic nuclei are going to be in this sort of position here. Okay, in blue. So they are nicely positioned above the optic chiasm. Okay, so that's the anatomy of the suprachiasmatic nucleus done. Let's now discuss the physiology of it. But I think we'll do that in the next video.